Okay, so I will get started. All right, so I'm going to talk about my research in Acadia, specifically looking at phenological changes in the plant communities in Acadia. And I did this through two different approaches. The first was hiking lots of mountains, and the second was with an experimental garden. And so if you joined our webinar last summer, I talked a bit about the hikes, the transects, to look at phenological change. But I don't think I actually got to the garden stuff at all. So today I'll be giving an update on our data and analysis from the transects and really digging into the garden work as well. And as you know, being signs of the seasons people, phenology is the timing of seasonal biological events. And ecologists such as myself are really interested in phenology because when we think about how climate change is affecting ecosystems and ecological processes, phenology, the timing of seasonal activities of animals and plants, is perhaps the simplest process in which to track changes in the ecology of species in response to climate change. It's something that's really visible and measurable, and it makes this kind of immediate connection to other people. So I feel in a lot of ways that I'm lucky as a researcher because it's easy to explain what I do when I'm sitting next to someone on a plane or if I just happen to meet up with friends and there's someone new in the group, it's easy to say that I look at plants and when they flower and leaf out, and often people will have really personal experiences tied into phenology, either from things like maple sugaring or their own gardens. They'll remember years in which they planted things and then there was a late frost and they lost all of that effort. And they'll remember years like 2012 when the spring was so early that things were just leafing out and blooming like crazy in February. And I used the back of this slide of 2016 as the hottest year so far because 2016 was the last year that I did this fieldwork in Acadia National Park. This was all part of my PhD dissertation. And so I was working in Acadia starting in 2011, but doing the fieldwork that I'll talk about in 2013, 2014, 2015, and 2016. So I just want to introduce you to the two big questions I'm looking at. The first, there we go. The first is how can we rapidly assess contemporary leaf out and flowering changes in a complex landscape? Anyone who's been on Mount Desert Island or hiked in Acadia knows that this is a pretty interesting landscape. You can go from sea level and be in a rocky intertidal area, and then within a couple miles and often under an hour, you can be on top of a mountain in basically a subalpine community. And so there's a lot of terrain complexity, a lot of elevational complexity, and a lot of vegetation complexity. And so trying to understand or how to really get a handle on what's happening across such a heterogeneous landscape, it's an intriguing question for land managers who want to get this kind of data and for ecologists who are interested in how do we approach something like this. And then my second question, I'm looking at what's driving this population level differences in leaf out responses within Acadia. So when I'm talking about interest-specific differences, those are differences within a species. So looking at low bush blueberry. And if you think about it, low bush blueberry, you don't just walk out your door at the end of April and there's a day where just boom, every low bush blueberry is leafing out. You'll notice some low bush blueberries leafing out early in the spring and some of them holding out until much later. And so is that a response to differences in local microclimates, or do some populations of low bush blueberry have different programming for when they are going to leaf out, or how much warmth or heat sum or spring weather they need before they'll leaf out? So those are the two questions I'll be addressing today. And I just wanted to take a moment to acknowledge how lucky I was to be able to do this work in Acadia National Park, where most of my dissertation research really looked like this. I got to spend time in a beautiful location hiking and sitting outside in gardens and writing things down in my notebook as I noticed them. So it was really an incredible experience, and I have a lot of people at Acadia National Park to thank, but specifically Abe Miller-Rushing, the science coordinator, and everyone at Resource Management who helped me out with this project. So I'm going to start with the hiking, the observational transects. And so this gets into the question, how can we rapidly assess contemporary leaf out and flowering changes in a complex landscape? And I asked this question because I came from a lab at Boston University that had focused on Thoreau's Concord in Massachusetts. And so this is our classic figure from the Concord data set, where on the x-axis, there's mean spring temperatures from cold springs all the way out to warm springs. And the y-axis goes from the 19th of April, so kind of early springs, up to late May. And you can see that there's a really clear relationship between spring temperature and mean first flowering date, where in warmer springs, things are flowering earlier, and in colder springs, things are flowering later. And this is this nice, simple linear regression gives us a number to put on that, where we can say for every degree Celsius that it's warmed, the spring or the first flowering date is advancing 3.2 days. And that's a negative 3.2 because it's advancing. It's happening earlier in the year. And so this was the lab that I was trained in as a grad student. And this is the kind of work that they had been doing in Concord, Massachusetts since the early 2000s. And it's really interesting work, but it depends on having a huge historical data set from someone like Henry David Thoreau. Because in order to make this figure where each individual dot on that figure represents 1 year, where you have the mean spring temperature for Concord in that year and the mean first flowering date for a suite of about 30 species in that year, this is the data behind that. So you can see that the years stretch from 1852 all the way up in this case to 2000, I think this is 2011, and the years continue to accumulate as the Premack lab continues to work in Concord. But to make that one figure took a lot of effort and a lot of data. And when I started my dissertation research in Acadia National Park, I didn't really have time as a new PhD student to put in 20, 25, 30 years of observing phenological change in Acadia National Park. I figured I had about 5 or 6 years as a PhD student. So I needed to come up with a way to rapidly assess leaf out and flowering times. And I was looking at the historical records for Acadia National Park, and there's a lot of historical botanical work from Acadia. It was this huge hotspot at the end of the 19th century for botanists to come and collect herbarium specimens. There's a beautiful flora that was written in 1890 or published in 1894 from fieldwork that dates back to the early 1880s. But there weren't phenological records. So all of these folks who came to Acadia to botanize, they were mostly wealthy elites from Boston and Philadelphia and New York. And they had the leisure time and they had the botanical training to do this as a hobby. And they would do this while they were summering. And by definition, summering means that they weren't here in the spring when things are actually leafing out and flowering. And the folks who lived on the island year round and may have been recording those observations, or maybe didn't have time to record those observations because they were working and trying to prepare the estates for these wealthy elites. If they did record them, they weren't archived. So I couldn't find historical records of leaf out and flowering phenology for Mount Desert Island. And basically I was starting from scratch and needed to come up with a way to get a lot of phenology data very quickly. And that's where the complexity of the landscape actually helped me out, because as you can see from this map of Mount Desert Island, there's a lot of complex terrain. There's a lot of elevation gradients. And anyone who's hiked Cadillac or Sargent or Pematic knows that when you start at the base of those mountains and you're wearing shorts and a t-shirt, by the time you get to the top, you've added a couple layers. You might be at the summit of Cadillac and you've put on a fleece and a hat. because you have just hiked through a bunch of different microclimates. And so you've actually experienced different types of springs as you've hiked from the low elevation trailhead up to the summits of these mountains. And so taking advantage of those microclimates and that elevation gradient, I could collect a lot of data in a single year by really breaking the mountain down into different zones. And so that's what I did. I was looking at 7 elevation zones on 3 mountains, Cadillac, Sargent, and Pematic. I monitored each mountain twice a week, hiking up the North Ridge Trail and down the South Ridge Trail, starting in early April and ending on June 30th. And June 30th was our cutoff because we were interested in spring flowering plants. And so we needed kind of a hard date to define our spring. I did this for 4 years, starting in 2013 and ending in 2016. Yes, that's correct. I looked at 30 species altogether, but I'm going to present data for the 9 most common species. So things like low bush blueberry that grow across that entire elevation gradient. We were able to collect a lot of data very rapidly, because if you have 3 mountains and 7 elevation zones, in a single year, you can look at 21 different populations of low bush blueberry, and so get 21 data points for that one species very quickly. And so this schematic kind of helps to show how I divided these mountains. At the top, you can see the trail signs for the 3 mountains, Sargent, Pematic, and Cadillac. And I've divided this imaginary, let's say it's Cadillac Mountain, into the north and south aspects, and then divided the elevation zones into different sites. So on the north, as you move through the first elevation zone, you're in site 1, and then you get into the second elevation zone at site 2. Across all of these mountains, I had my cutoffs as 0 to 600 feet of elevation, then 600 to 900 feet of elevation. Site 3 went from 900 feet to 1,200 feet, and site 4, which is the summit, went from 1,200 to the summit, and then those exact same boundaries down the south side. And so that was how I basically sliced these mountains into different elevation zones. And I wanted to see how the microclimate changed as you moved across these elevation zones. And so I put out temperature loggers. The big picture with the hand shows what these little loggers look like. They're called pendant loggers, and I hung them from corner gutter pieces, that white item in the second picture with the little tag on it was the shield, so that they wouldn't be exposed to full sun and then experience temperature spikes. So the corner gutter pieces that are upside down allow air to move through the corner gutter piece, but protect the pendant from full sunlight. And these were hidden off the trail. I had 1 in each of those elevation zones on the North Ridge side and 1 in each elevation zone on the South Ridge side, for a total of 7 different loggers per site. And these were great. I remember going into Home Depot when I was putting these together and buying 21 corner gutter pieces. And the really nice gentleman in Ellsworth who was helping me out had a very confused look on his face. And he said, "What shape is your house?" But they turned out to be a really cheap and effective sunshade. And we hung the pendant loggers from the middle of the corner gutter piece with a zip tie. We drilled a hole in the top and zip tied it in with a tag that said, "If you found this, please don't move it. It's part of Acadia National Park research." And there was contact information for the Resource Management Division. So those recorded our temperatures, and we were specifically looking at spring temperatures. So we wanted to know what was happening in March and April. And so to give you a little insight into how this worked out, it turns out that it's not as simple as saying it's cold at the summit and it's warm on the southern aspect of these mountains, and the lowest elevations are the warmest spots. So to show that, I'm just going to fill in the Cadillac 2016 mean spring temperatures. These are March and April temperatures. So the summit was the coolest spot that spring, but then it kind of bounced around, and it turned out that on the North Ridge, on the lowest elevation site, it was really warm And if you've hiked the North Ridge of Cadillac, you know there are some of those open, exposed, rocky areas, and you go through some really kind of like warm pine forests. But on the South Ridge side, if you've hiked the Cadillac South Ridge Trail recently, you start off by hiking through about a mile of a just deep, closed canopy conifer forest where it's really cool. And so the microclimate is really complex when you think about where the coolest and warmest spots on the mountain are. And so for the rest of my analysis, you're going to kind of forget about where you are on the mountain, and we're just going to match up the phenology observations with the mean spring temperatures. So I'll be showing my results on a figure that looks like what I showed from Concord, Massachusetts. The x-axis, we have mean spring temperatures. These go from 0 to 3.5 degrees Celsius. And the y-axis goes from, mid-April through mid-June for the date of leaf out. And in this case, instead of having each data point represent a year where we have a mean spring temperature for all of Concord in that 1 year, and then a mean leaf out date for all of the species in that year, I'll have a data point that represents a single elevation on a mountain with an aspect and a single species. So a data point will represent, for example, the summit of Cadillac for low bush blueberries. So it's going to look a little bit crazy for a second, and I'll try to talk you through that. Here's all of the data. We had temperature data for 2014, 2015, and 2016. It took us a little bit longer than expected to get those temperature loggers deployed. So this is 3 years of data across 21 sites. I put up all of the data for the 9 species, the 9 most common species. At the top of the figure, there's photographs of 6 species. And those are the 6 species for which we have significant relationships between leaf out and mean spring temperature. So I'll just read across the top. The first picture is huckleberry. The second one is sheep floral, Canada mayflower, excuse me, 3-2 cinquefoil, low bush blueberry, and wild raisin. And so the 6 lines that are visible in the figure represent the regression lines for those 6 species. So when we look at those, it can be a little bit overwhelming as just kind of a giant plot of lines, but I'll just let you know that in each case, they're leafing out earlier in warmer years. And even for the 3 species that did not have a significant relationship, that pattern remains true. And so, for example, the wild raisin is leafing out 6.1 days earlier for each degree Celsius that it warms. So the regression coefficient on that slope is 6.1 or negative 6.1. And the huckleberry is leafing out 1.7 days earlier for each degree Celsius that it warms. So huckleberry has that significant relationship, but it's not as dramatic as the wild raisin. So I'm going to show the same type of figure for a flowering date. These are the same 9 species. Across the top, we have 6 species that have a significant relationship between flowering and mean spring temperatures. Here's all of that data once again. The 6 species in this case are chokeberry, bunchberry, huckleberry, sheep floral, 3-2 cinquefoil, and low bush blueberry. And in this case, those slopes range from 3.6 days earlier. So that's the chokeberry, it's flowering 3.6 days earlier for each degree Celsius increase in temperature. And sheep floral represents the smallest regression coefficient at 1.8 days earlier for each degree Celsius that it's warming. And again, every single species is showing this pattern. These are the 6 with significant relationships. And when we zoom in on that to look at the difference between Mount Desert Island and Concord, we have the same pattern. They're all advancing their phenology in response to warmer temperature. But for these 4 species that we monitored both in Concord and on Mount Desert Island, the magnitude of that shift is greater in Concord across the board. And that's true also for flowering species. There are, again, 4 species that we monitored on Mount Desert Island and in Concord for flowering. And again, they're all shifting earlier in response to warmer temperatures, but the magnitude of that shift is greater in Concord, Massachusetts. So we were able to look at changes in phenology in response to different temperature regimes, but that required having a person like myself or some of my undergraduates from College of the Atlantic hiking 3 mountains twice a week. So there was a lot of hiking happening to record all this data. And we really wanted this kind of work to continue to happen. And phenology monitoring is, as you know, at Science of Seasons, very important to track changes in how plants are responding. So a second part of this analysis was to look at different monitoring strategies. So what would happen if in the future we were only able to keep up this kind of work on Cadillac or only on Sargent and Pematic? What would happen if we only looked at the southern aspects of these mountains or the northern aspects of these mountains or just the low elevation sites or just the high elevation sites? Or what if we did this for fewer years or more years? And so we took the most common species, and that was lowbush blueberry, the one that we had the most observations for across our 3 years of data. And we looked at what would happen if we subset the data that we had collected over those 3 years into those different categories. So we calculated what that change in phenology per degree Celsius would be. So like what the slope would look like along with the standard error. So in this figure, you can see the estimated flowering response. The original model refers to what happens with all of that data. So for flowering lowbush blueberry, that's a negative 2.8 regression coefficient, or it's advancing 2.8 days per degree Celsius. And if we broke it down by mountain and looked at just Cadillac or just Sargent and Pematic, Cadillac and Pematic, Cadillac and Sargent, you can see that that regression coefficient moves around. And when we take out the Cadillac data and just look at data from Sargent and Pematic, that regression is no longer significant. That's why that little bar is now red. So Cadillac is important in terms of being able to provide enough data across enough microclimates to get a good estimate of how flowering is responding to temperature. We did the same thing looking at aspect, so south aspect versus north aspect. And you can see again that the north aspect only is not significant. So it didn't cover enough of a range of microclimates. It didn't get enough data just from that north aspect to give us a good estimate of the flowering response. And then if we look at high elevations only or low elevations only, again, we see this where the high elevation is not significant. To get a good estimate, the low elevations did an okay job, but that spread on the standard error is pretty large. And then if we only looked at 2 years of data, just 2014 and 2015, it's a significant estimate, but that standard error is very large compared to our original model, which incorporates 2014, 2015, and 2016. So we did that same kind of subsetting for leaf out response. And in this case, again, looking at just lowbush blueberry, the complete model was had a regression coefficient of negative 4.3, or it's advancing 4.3 days for each degree Celsius. When we subset the mountains, we actually have a pretty nice estimate across the board for leaf out. And I think that's because we have slightly more leaf out data than flowering data, because every plant that we monitored was leafing out, but not every plant that leaves out produces flowers. When we look at the north aspect and the south aspect and break those down, we still get significant models, but you can see that they differ. So the north aspect is kind of underestimating that change in leaf out response, and the south aspect only overestimates that response. And you see that same split with high elevations and low elevations, where with just the high elevation data, you get an underestimation, and the low elevations overestimate that response of leaf out. And if we only have 2 years of data, we don't have a significant model at all for our leaf out response. We really need those 3 years of data to get a good handle on how leaf out is changing in response to temperature. So the main conclusions from this hiking and observational transect approach was that we have leaf out and flowering phenology that's responsive to temperature across these environmental gradients here in Acadia. And the shift in days per degree Celsius is smaller than in Concord, Massachusetts. So they're advancing just like they are in Concord, but they're not advancing as much. And that's even on a species by species level. So the same species growing in Concord seems to be shifting its flowering or leaf out more dramatically than on Mount Desert Island. And we recommend monitoring strategies going forward that focus on Cadillac, or at least continue to include Cadillac, southern aspects, and definitely more years. We want to keep monitoring these plants in Acadia National Park into the future. And I'll talk, I think, at the end a little bit more about our plans for that. So the second half of this talk is about experimental gardens. And with this, I wanted to ask or answer the question, what's driving population differences in leaf out responses within Acadia National Park? Because as we were doing the hiking to monitor plants across 3 mountains in Acadia, it was really obvious that there are population level differences in phenology. A lot of species, especially those 9 common species that I just showed you, grow across that entire environmental gradient, and they leaf out and flower at different times at different places on those mountains. So is this a population level genetic difference? Are the plants on top of Cadillac, the sheep's laurel on top of Cadillac, just genetically different from the sheep's laurel at the base of Cadillac? Or is it just a simple response to local environment where it's just colder on the summit and it's warmer in another place, and the microclimate is driving the timing of leaf out and flowering? And there's good reasons for either hypothesis. So population level differences could be an adaptation if you are a plant that's growing in a harsh and unpredictable environment like the summit of a mountain, because you might be more likely to experience a late season frost, which means that you might be more reluctant to start leafing out or flowering at the first sign of warmth, because you know it could be kind of a false start. And so you might have some kind of local adaptation to be more cautious, whereas plants in lower elevations, especially in areas where they're maybe not as exposed to things like wind or ice storms, you can have that exact same species, but it's predisposed to leaf out or flower as soon as possible. So it can start growing and reproducing and accumulating energy. Because we know in Maine, everything has a pretty short growing season. So you want to extend that growing season as much as you can. And so if you have trade-offs of, do you want to extend the growing season and get started as early as possible, or are you nervous about a late season frost, different populations might weigh those trade-offs differently. And that was a huge anthropomorphizing of plants, but I think that it really drives home the question of why there might be local adaptations. So to get at that question, I put together a common garden experiment. And common gardens are a pretty classic experimental design where you transplant plants to different locations. And so we had 3 gardens. There was a garden at the summit, the mid elevation, and the low elevation on Cadillac. This was off of the Auto Road. And we filled them with plants from those 3 elevations. So if you imagine that my beautiful drawings here are all sheep's laurel, and the purple sheep's laurel are the summit population. We put purple summit sheep's laurel into all 3 gardens. And the green sheep's laurel are the mid-elevation gardens, or the mid-elevation sheep's laurel, sorry. And we put green mid-elevation sheep's laurel into all 3 gardens. And we did the same with the orange low elevation sheep's laurel. So within any one of these gardens, if we looked at the low elevation garden, those plants are all experiencing the same local microclimate. So if they all leaf out at the same time, regardless of their source population, then you know it's the local environment that's driving that leaf out. But if within one of those gardens at the low elevation, you have leaf out occurring at different times, all 3 of those plants are experiencing the same local climate, but they're responding to it differently based on whatever local adaptation they had from their source population. So this is a nice photo montage of how we actually did the garden building and construction. I worked with Friends of Acadia volunteers who were incredibly generous with their time and their skills. And we were able to put these gardens together over a 2-week period in the fall of 2013. And so we cleared these sites. One is at the base of Cadillac, one's at the mid-elevation around one of the pullouts, and one is at the summit of Cadillac. And then we were able to dig out plants from each of those 3 sites and plant them into the gardens. So each garden has sheep's laurel from all 3 elevations, low bush blueberry from all 3 elevations, and 3-tooth cinquefoil from all 3 elevations. And we also tagged those 3 species around the gardens that were not transplanted to see how local plants in the same local environment as the gardens, but without the potential trauma of being transplanted, would respond to the local environment in terms of their leaf out. And we actually did monitor leaf out and flowering over 2014, 2015, and 2016, but not enough of the individuals in the gardens flowered for us to do a statistical analysis of flowering. So this is really focused in on leaf out phenology. And so our hypothesis that population level adaptations in phenology would exist across Cadillac's elevation gradient. If that hypothesis was true, we would expect the source population, highlighted in yellow, to be really important. And so in this chart, I have the 3 species. The top one is sheep's laurel, then there's low bush blueberry, and then 3-tooth cinquefoil. And then along the top row, I have the different factors that could be driving leaf out phenology. So the garden site, that's the local environment, highlighted in orange. Year, which is actually also a good proxy for a local environment, because from year to year, the environment will shift and change. We know that there are some really warm years and some really cold years and weird years like this year where there's winter for a long time and then all of a sudden it's summer. And we have the source population, and then the last 3 columns represent interaction effects, where there's some kind of interaction between the year and the source population, or the site of the garden and the year, or the site and the source population. And so our analysis was to look species by species at which of these different factors was most important in driving the date of leaf out. And so here's our results. The stars represent the factors that were significant in individual species level leaf out. And the 3 stars show things that are really significant. So across the board for all 3 species, the garden site and the year, those 2 factors that are related to local climate, were very important. Source population on its own was only significant for low bush blueberry. So there's evidence that there's local adaptation, but it's not as important as the garden site, the local environment. For sheep's laurel and for 3-tooth cinquefoil, that source population is important as an interaction effect. So it either interacts with the site or with the year in the case of sheep's laurel. And so there is, again, evidence that there is something happening at the source, that local adaptation from whatever source it came from. But it's just not as important and not as consistent as the local environment. So the local environment is really driving that leaf out phenology for all 3 species. And this is actually kind of good news, because from other studies, we've seen that plants that can shift their phenology in response to different temperature cues actually tend to do better, have higher fitness, are less likely to go locally extinct than species that can't do that. And so showing that all 3 populations of these species can shift their their leaf out time in response to different climates shows that they might be more resilient to climate change in the future than we might have thought. So the summit populations of these 3 species will be able to adapt to a warmer future. Because if you think about it, this experiment is also kind of a low-tech warming experiment. So we didn't have to bring out heat lamps or do fancy shoveling around our sites. We basically just took plants from the summit and put them in the mid-elevation and the low-elevation sites. And those sites were warmer than the summits. So we did a low-tech warming experiment and found that these plants were able to respond pretty well. And we had very low mortality across the board. So not only could they survive in the warmer environment, but they seemed to also be able to shift their leaf out to accommodate that warmer environment. So we have leaf out phenology cued by local microclimate, regardless of the source population. And these population level adaptations in phenology are just not important on Cadillac Mountain for these 3 species. And our high elevation populations, those summit populations, didn't display reduced plasticity or the inability to shift their phenology compared to low elevation populations. And that's an interesting finding, because in other places where they've done common garden experiments like this, in the Alps, in the Pyrenees, in the Rockies, really big mountains with really large elevation gradients, that's a pretty common result to see high elevation populations have less of a response to warming than low elevation populations. And here we showed that across a really compressed environmental gradient in Cadillac, we just don't see that. So perhaps there's enough genetic mixing across these sites. You know, it's not that far from the base of Cadillac to the top of Cadillac. A pollinator could probably make it that far. They're not isolated enough to develop some kind of genetic adaptation. So overall, we have these ridge transects on Mount Desert Island demonstrating advancing plant phenology in response to spring temperatures in Maine. And the phenological responses in Maine match the direction, but not the magnitude recorded in southern New England. So in Concord, Massachusetts, they're advancing at a much faster rate. And site conditions are the most important factor cueing leaf out on Cadillac Mountain for our 3 species, sheep's floral, low bush blueberry, and 3-2 cinquefoil. So those are the big conclusions from these 2 studies. And I just want to point out that I'm still working in Acadia National Park, and I'm actually still working with some Thoreau data. So I'd be happy to answer any questions about this. I have one project that's actually looking back at that Concord data set to look at changes in leaf out times and changes in flowering times since the 19th century, where we show that leaf out is actually responding faster than flowering times, which means that the canopy is closing earlier on understory species that rely on having a high light environment before the canopy closes. So if the understory species are not keeping up with their phenology, they're actually losing the timing of that light environment. So they're getting less light in the spring, which could affect their ability to take up carbon or to reproduce. I'm also continuing some work in Acadia National Park. That's a picture of Sargent Mountain Pond, which I cored in September 2017 to look at pollen captured in the lake sediments there, to look at what's been happening around Sargent Mountain Pond since glaciers retreated 16,000 years ago. And so I haven't actually looked at the pollen captured in that core yet, but hopefully soon I'll be able to tell you about how vegetation and communities around Sargent Mountain Pond have changed over the last 15,000 years. So with that, I'd just like to acknowledge some of the amazing people I worked with before I jump into questions. The first photo is my lab mates, Amanda Gallinat, myself, and Lucy Ziff, who are all members of the pre-Mac lab. They came up to Maine to help out with some field work, and they were just really excellent people to have around when I was working in Boston. Then Abe Miller Rushing, the science coordinator at Acadia National Park. He was on my dissertation committee and was just a really invaluable resource while I was doing work in Acadia National Park. And then in the last photo, Richard Premack, my advisor, is hiding behind Lucy, and he was an excellent advisor at Boston University. I also have 3 undergrads who did a lot of work on these projects, but didn't take any beautiful pictures with me, or I didn't fail to take pictures of them. Paul Excoffier worked in the gardens the very first year that the gardens were set up, so he was the real guinea pig and did a wonderful job monitoring those gardens. And then I worked with Ella Samuel and Natasha Krell from College of the Atlantic in 2015, and Ella Samuel came back in 2016. And they worked on both the gardens and the transects, and it was really wonderful to work with them. Across the bottom, I have funders who helped to fund this research from the Park Service, Schoodic Institute, National Science Foundation, New England Botanical Club, and the Waterman Fund. And none of this would be possible without those resources and their support. So thank you very much, and happy to talk more.
Awesome. Thank you so much, Caitlin. That was a really wonderful presentation with lots of really beautiful photos and great imagery and figures and everything. So I really appreciate you sharing that with us. It's great to see all that research coming together.
Yeah.
So just so everyone who's tuning in, a quick reminder, if you move your cursor around on your window that you're viewing through Zoom, you should have a little menu at the bottom. And if you click on the chat icon, then you should have a little box that you can type in a message, either addressed to everyone, or you can send it to me, Elizabeth. And start filling in your questions there, and I'll read them out as they pop up in the chat box. While we wait for any questions to come in, Caitlin, I actually have a question for you that I was thinking about.
Georgia asked, sorry I missed that before. I can either go back, let's see if that will work. Can try to get back to these figures, but I also have the regression coefficients for this. Here we go. That's leaf out. So with leaf out, the Canada Mayflower was leafing out 2.5 days earlier per degree Celsius, and that, which color is that one? I'm not sure I'm going to be, I don't have the legend on, yeah, I don't have the legend on this figure, but I have the legend on the figure. So this paper is actually in review right now at Ecosphere, and I'll send out everything as it becomes published to Science of the Seasons. But we'll actually, one of the reviews had us mixing up this figure a little bit and making it easier to read. So sorry, I can't point out exact colors on it. But for the flat date of flowering, I can say that Canada Mayflower was not significant in this case. So the trend for Canada Mayflower is that it was blooming earlier in warmer years, but it was not a significant linear regression with the data that we had. And I think part of this, especially after doing the analysis looking at low bush blueberry under different monitoring strategies, I think part of that is just the reduced number of data points, especially if we look at our mean spring temperatures here. From 0 to 3.5 is not a huge spread. With the Concord data set, which I can actually kind of split back to, because they had so many years across such a huge range from the 1850s through the 2000s, their mean spring temperature spread goes from 4 degrees Celsius to 11 degrees Celsius. So they're covering a much wider range of spring temperatures. And at one point, one of my lab mates told me that I should rename my thesis to be Phenology in Acadia National Park over 4 normal years, because I started collecting this data after 2012. So I missed the really early, crazy warm year and started collecting data in 2013. And when I looked at the temperature data, which I had from 2014, 2015, and 2016, when I looked at that temperature data for March and April, it's actually pretty similar to each other. So even though 2015 had that weird snowpocalypse, and there was still snow on the ground in Acadia when I started my work, once that snow melted, everything just kind of went back on track in terms of being a pretty normal year. And so the mean spring temperatures were not that different from the 2 other years, which is why we need to collect more data.
Do you think that there's other environmental conditions that are playing a role in the difference between MDI and Concord, like maybe like precipitation and last frost date and those kind of things? Like how do you think about those kind of things as well, and how they may play a role?
Yeah, so I think being a maritime climate where I think that having Concord as a kind of sister site in some ways is nice. There's a lot of overlap. There's about 300 species of plants that are in both locations, but they're both very different places. Concord has had the urban heat island effect from being a Boston suburb just really kind of jack up the temperature in a way that we don't experience on Mount Desert Island. So our temperatures are warmer than they were in the 19th century, but they're not dramatically as much, or it hasn't changed as much as in Concord, Massachusetts. So Concord's kind of like a canary in a coal mine in terms of like getting an example of what rapid climate change looks like from just the urban heat island effect. So I think there's a little bit of that where Concord has experienced more warming than Mount Desert Island. I think the climate is definitely different being inland versus being on the coast. Concord is actually pretty well conserved landscape in a lot of ways. I think something like 2/3 of the land in Concord is under some kind of protection. But there's still a lot of development pressure in Concord. So that means that a lot of the plants that Thoreau was looking at, those populations have declined in abundance. Some of them have gone extinct, which also might be affecting what we see in terms of the phenology. Because with Concord, and we did this as well on MDI, we were looking at the date of first flower and first leaf out. And that's in part just a relic of how 19th century naturalists were doing it, because those were the things they got excited about. And they didn't have the systematic peak flowering date or counting inflorescence that could allow a metric that wouldn't be as skewed by the abundance of a species. I think there's probably a lot of reasons why Concord is different from Mount Desert Island, and some of them we could try to tease apart with metrics like looking at precipitation and looking at things like growing degree days instead of mean spring temperatures, and really getting into some of the nitty-gritty meteorological stuff. But some of the things I think might be much harder to resolve, just in terms of Concord's history and the fact that the Concord data set is just bigger and older and based on a much longer time span that covers a wider range of temperature or spring temperature experiences.
Is Acadia and the research that they're doing, or just the kind of planning and continued monitoring that they do there, do you have a sense of like how they may be able to use the outcomes of your research?
Yeah, so we've started talking about using phenology as a trait in climate change vulnerability assessments. And so of the 9 species that we looked at, there was one species, starflower, that didn't have a significant relationship between leaf out or flowering and mean spring temperature. And that might be a sign that it's less responsive to changes in temperature and might be more vulnerable to climate change than some of the other species, which had at least one phenophase that was responsive to temperature. So starting to use that data in terms of building up a database of phenological traits. We've also looked at building this into their phenology trails, which have been really active on the Scudic side of Acadia, and we want to bring that into the MDI side, potentially through Nature's Notebook, the USA NPN app, or with iNaturalist to get a kind of cast a wider net on people making observational records as they hike. So that's kind of still in the early planning stages in terms of continuing this phenological monitoring program.
Right, and kind of on that same, not to interrupt you, I'm sorry.
Yeah, go for it.
On that same thought process. So the Coastal Maine Botanical Gardens, we, as Signs of the Seasons, have been working with them this year to establish some long-term monitoring sites in the gardens. And one of the species that they're focusing on is low bush blueberries. So that maybe that's an area where there could be some comparison with Acadia and Scudic.
That'd be great. The gardens, I was about to say, have actually been repurposed for more experimental studies with Nick Fisichella at Scudic Institute, where the physical gardens remain. They're actually really beautifully built and made out of cedar, so they're going to be there for a while. But they've taken the plants out. Actually, the plants in the Summit Garden were used in some of the Summit restoration on Cadillac, which is kind of cool to think about. But they're now full of tree seedlings, both from locally sourced within Acadia or from the island and from more southern regions to look at potential for managed relocation and assisted migration to see how different tree species respond to this range of climates. And it's actually expanded so that it's not just the 3 gardens on Cadillac. There's now a set of gardens at Scudic. And as I understand it, I think schools across Maine are also building similar gardens to continue this common garden experiment across a huge geographical area, which would be really cool to see what Nick finds with his studies.
Yeah, that's really amazing. Cool. Well, I don't see any other questions coming in through the chat. I think, Esperanza, is that you signing in? Or
yeah, I was just gonna say if there aren't any other questions, I would just thank you again, Caitlin. This is really fascinating work, and it's happening right here in Maine. So we'll be sending this out, far and wide for people to be able to take a look at this research. And I hope to make some connections. We've been talking with Acadia Scudic and about the new work that's going on, and we have a lot of data actually of probably some of the tree species and so forth. So stay tuned.
Great, that sounds awesome. And I mentioned that the Transect paper is in review, but the garden paper is actually about to be published. So I will send that out to you as soon as it comes out in the American Journal of Botany. And both of these papers will be open access. That is another thing that I should definitely thank the funders for, because it's huge that we're able to get these into academic journals, but also available so that anyone can read them. They're not going to be hidden behind a paywall. So I'll share those links as soon as they come out. And the garden paper should be out later this month.
That's excellent. Thank you so much. That's going to be a great resource to have. So I want to thank everybody for participating. And if anything comes up, I'm sure we could get other questions to Caitlin if necessary. So Thank you all.
Thank you so much.